speaker today. For many of you in the room know him or know of him. Pat Schloss is the Frederick Noby Collegiate Professor of Microbiome Research at the University of Michigan. And yeah, so Pat is, I'm sure many of you know, for developing the mother pipeline and does a lot of microbial ecology work. He is the chair of the Journal of ASM. That's the proper title for that, something like that. And you will probably be familiar with a number of publications, which tend to be rather highly cited, over 1,000 citations to his work. And the original mother paper, I think, is creeping up on 10,000. So I don't know if you've got a celebration plan for that when that comes around. But this is all only Pat's day job. He also runs a farm with his family. And I can tell that he's, as a Canadian, I can tell he's raising his children right because they're currently constructing a hospital. So with that,
thank you. So it's great to be here with you all. This is, I think, my third time being here professionally. So I actually grew up to some extent in State College and elementary school, 1st through 5th grade. And so it's cool to kind of see how much campus has changed as much as I remember from being this little tyke. And to put me in context for you, an anecdote I'll tell you is that my dad is a professor in special education here. I remember being in his office, this was like mid-80s, and he had this thing that he put his phone on and allowed him to pull up the library card catalog, whatever. I think that's just like, it's like internet. And I think it was the stupidest thing. The library's right across the street, Dad. Why don't you just go over there? And of course now, like, the library closest to me at the University of Michigan has no books. And so don't come to me for forward-looking visionary statements because I clearly don't have what it takes. But it's great to be here and spend the day with so many great people doing really diverse things. In thinking of the microbiome, I think it's really true sense of being very broad, not just humans or guts, but soils, corals, everything that's out there. And so hopefully you can take what I'm saying today from what I'm approaching from a human-centric, mouse-centric worldview and see how you might think about it in your own system. Always happy to talk with people about what we're doing. So great. And so what I'd like to do is share with you my sense, as the title says, of how we understand disease and think about it through the lens of the microbiome. And you don't need to know this if you're in the microbiome center, but the microbiome is this collection of microbiota that live on you. Everything from bacteria, archaea, microbial eukaryotes, viruses, live in and on the human body. Somehow I've recruited 3 virologists to be postdocs in my lab over the last several years. They talk about things I have no idea. And I try to say words like age, and they thankfully don't sneer at me like you use the wrong word. So trying to think more expansively than just bacteria when we think about the human microbiome. The numbers are really squishy. Everything from 1-to-1 bacteria to human cells to 10-to-1. Many microbial genes relative to human genes. And the numbers really aren't that important. But the numbers, I think, are kind of inflated in some cases to emphasize to people that there's a lot there. There's a lot of diversity. And hopefully you know that whether it's 10 to the 12 or 10 to the 14 or 10 to the 15, who cares? That's a really freaking large number, right? And so there's a lot of diversity and a lot going on. And so we think that because there's so many bacteria, so many viruses, on, that they're really important in health and disease. And so what do we know so far? Well, I think initially we thought a lot more, and now we're kind of revising and perhaps drawing brighter lines around what we really do know. But we know that, we look at germ-free mice and compare them to conventionally raised mice. We see that bacteria are really important for digestion, epithelial health, developing the immune system, thinking about colonization resistance, that the gut bacteria, for example, are doing something to prevent every pathogen from colonizing our gut and making us sick. And so these associations with disease then, I think, and maybe I'm a bit bearish on the microbiome, but I feel like there's still a lot of conjecture that kind of connecting the microbiome in terms of, when I say it today, we think of the gut, to every disease under the sun, that maybe we need to be a bit more cautious in how we make those And so things that we do feel like there's a stronger association or perhaps mechanistic link to include things like diabetes, inflammatory bowel diseases like colitis, Crohn's disease, colonization resistance, and colorectal cancer. And so two diseases that I'm going to talk about today with you are colonization resistance in terms of resistance to Clostridium difficile, which is largely a nosocomial infection that people get in the hospital when they go on antibiotics, and then colorectal cancer, which is unfortunately fairly common cancer in the US and elsewhere. And so
why do my headphones not work?
And so I like to share a quote with people who talk about the microbiome to get them thinking really about how we do our science. And this is a quote from a colorful individual in my field where they said, it would appear to be a pointless and doubtful exercise to examine and disentangle the apparently random appearing bacteria in normal feces and the a situation that seems controlled by 1,000 coincidences. Right? So why do I have my gut microbiota? Why do you have yours? Well, there's 1,000 coincidences that could explain it. Yet, I have nevertheless devoted myself now for a whole year, virtually exclusively to this special study. It was with the conviction that the accurate knowledge of these conditions is essential for the understanding of not only the physiology of digestion, but also the pathology and therapy of microbial intestinal diseases. So this is the human microbiome in a very somewhat arrogant, naive quote. Right? So any ideas who may have said this? Thoughts. Come on, there's no dumb answer. So I'll dispense with the suspense, but this came from Theodor Escherich in 1885. Okay, German pediatrician who had parents bringing kids to him with diarrhea, wanting to know why they had diarrhea. So what I like this quote is because this is 1885. People are still writing the same paper, basically, 130, 140 years later. And I like to challenge people to think about what do we know that good Dr. Escherich didn't, right? So he didn't know that he had Escherichia coli in his gut, right? He didn't know about the so-called great plate calonoma, that most bacteria in us aren't readily culturable. 1885, Darwin had just kind of gotten on Hegel, proposed evolution, right? Hegel had just started thinking about Darwin's work in context of ecology. There's really no formalized statistics. DNA was a molecule of inheritance, right? On and on and on and on, the things he didn't know, but he was able to figure out. So here we are now, 2018. Thing I say to administrators is, it's 2018, we should be able to do this, right? And so it's 2018, we should be able to do this. But it's hard, right? And when kind of what we've learned over the last 30 years, so it's hard, it's really complicated, there's a lot going on. But this is like, I think, the mission statement for a lot of microbiome research. And again, whether you're studying infant guts like he was, corals or soils or biofilms at the bottom of a lake or the ocean, we're trying to disentangle these 1,000 coincidences to understand how they relate to physiology, pathology, and then thinking about therapy. And so I like to think about broad mechanisms. And people, I live in a med school where everyone's like, what's the mechanism? What's the mechanism? What's the mechanism? And I'm an engineer by training, and so I don't really think about mechanisms. Instead, I think maybe more about processes or how can we control or engineer things. And so I like to think about these broad mechanisms or broad ideas about how the microbiome might be involved in disease. And so, again, as an engineer, I've never taken a lot of the training that microbiologists take, like say intro micro. And so I had the benefit of Joe Hamilton, my postdoc advisor, teaching me Koch's postulates. I think every freshman or sophomore in college learns. But the idea that there's a single bug that causes disease, right? So E. coli causes a urinary tract infection. Bacillus anthracis causes anthrax. And I think we're still very much in that mindset with the microbiome. What is the bug that causes colon cancer? What are we missing to let C. difficile colon? Right? But it's always kind of this single bug, single disease. And hopefully what we're learning from microbiome research is that this is not the case. That there can be many bugs causing disease. And so to me, this is very much the Koch's model approach is very much like the simple model of the genome of Mendelian inheritance, that we have a single locus that has perhaps a couple alleles, and then we get phenotypic variation, but it's that's being controlled by a single gene. And so I think what geneticists have been figuring out is that this is not really the case with most traits in the human genome or any genome, that there are many genes that can be causing the traits we see, and that many genotypes perhaps can give rise to the same phenotype, perhaps many combinations of these alleles or many combinations of genotypes can give rise to different types of pathogenicity. And so that there's a lot of variation, that it's not as simple as one bug or one gene causing disease, but combinations, or perhaps lots of combinations. Right? So oftentimes we think about one bug causing the disease, but perhaps it's the missing of loss of that bug causing disease. And added to this then is that many of our traits are complex. That this is one of my favorite data visualizations from something got cropped off, but this is a picture of cadets at some military school from like 1918. And so you'll see that the median height of these men, young men, about 5'7". Right? This was 100 years ago. So our genetics haven't changed so much in the last 100 years to make men taller. But no doubt our environment, how we respond to disease, nutrition, things like that have raised our heights. And so that while there's probably propensity to be short, it's baked into our genes, there's also environmental factors that are shaping our height. And so too with our gut microbiome, the diseases that might be driven from the microbiome, that there might be community structures that make you susceptible to something. But if you're never exposed to that something, then you'll never get a certain disease, or you'll never become obese, or you'll never get lean, or all these different combinations. That it's far more complicated than just who's there or what genes are present. But there's also these interactions to make these processes very complicated. And so I like to think of polymicrobial traits. You can see where I'm going, that again, we can have many different types of bacteria that perhaps have the same functions in the community that are driving health or driving disease. And in my lab, we like to think about, as I said earlier, colorectal cancer and resistance to Clostridium difficile infection. And so when people say to me, like, what's the mechanism? Like, there's many mechanisms, right? Which doesn't get me a lot of friends necessarily in the med school. But over 9 years, my students have kind of figured out how to deal with our more mechanistic reductionist colleagues. It's a challenge. It's a community that we're thinking about as being the driver of health and disease. So again, what are these broad mechanisms that drive microbiome-mediated diseases? And the first example I'm going to share with you is colorectal cancer. And a lot of this work has been done in collaboration with one of your all's kind of new colleagues, Matt Ruffin, who might even be on the call. He was in family medicine at Michigan and is now at Hershey as their department chair. And so we've got a really strong group of clinicians at Michigan that have been really instrumental in helping us to think about colorectal cancer and the possible role that the microbiome might play, and how we can use the microbiome to think about improving outcomes from colon cancer. So it's the third most diagnosed and second deadliest malignancy worldwide. Every 3.5 minutes, someone is diagnosed with colorectal cancer. And we don't know why most people get colorectal cancer. There are genetic causes, there might be dietary causes, but for most part, 70 to 85% of the cases have no known etiology. And the incidence and death rates are increasing with age, so about 90% of new cases and 94% of deaths occur in individuals 50 and over. So, but at the same time, that age is falling. And so I think the AACR released recommendations in the last year suggesting that younger people should be getting screening for. So that disease, and it's unfortunately very common, and seems to be becoming more common earlier. And so this is my moment in my public service announcement to say that if you're between the ages of 50 and 75, get screened for colon cancer. There are non-invasive tests, there are more invasive tests. There's trade-offs. You should talk to your clinician. Someone you love is 50 to 75. Call them tonight after my talk and tell them to get screened if they're ready. Unfortunately, my mom died at the age of 50 of colon cancer, and so I'm on kind of heightened surveillance. And so I've had a colonoscopy at 2, actually. They're not, yeah, the drugs are great. It's not, leading up to the drugs, you got to do it, and it helps, and do it for the people that care about you. And so the fact is that most people don't enjoy. So most people skip their screening, and that's why I try to make a point of encouraging people to get screened, because only about 1/3 of people will follow the recommendations for colon cancer screening. And in large part, that's because the colonoscopy starts with a bowel prep, where you basically flush out your entire gastrointestinal. It's like a really salty, bad Gatorade that you just have to chug like a gallon of. It's not fun. And it's also expensive. But it's kind of considered the gold standard. That's like the one way to really see what's going on in your colon. And so others have been developing non-invasive tests to look at, say, blood markers or stool markers for evidence of an adenoma or a carcinoma. So progression is kind of normal adenoma carcinoma in the colon. And so what we want to be able to detect are adenomas, because these are the early stage tumors that if you get a colonoscopy, one of the advantages of colonoscopy is that the gastroenterologist sees a polyp, they can remove it and you treat it. Whereas if you take something like FIT, fecal immunochemical test, they're looking for blood in your stool sample. And so it's not a treat, it's a diagnostic for screening, but it's not something that can be used to immediately treat like you could with a colonoscopy. And so there's these trade-offs between costs and sensitivity and specificity as well. And so one of the ideas we had was, can we take some of these kind of non-invasive tests and add to that microbiome information to improve our ability to detect earlier stage tumors and perhaps even later stage tumors. But again, the goal is to really be able to detect this disease earlier than later. And so Caitlin Flynn, a former postdoc in my lab, and others in our group put together kind of a model of how we're thinking about the role of the microbiome in colon cancer. And that the general idea is that there's some type of initial results to the colon, some type of mutation, perhaps on the host side, and that then microbes are involved in forming biofilms, interacting with the immune system to ratchet up inflammation and then to drive tumorigenesis. And kind of one of the things that people tend to see is a preponderance of bacteria like Fusobacterium nucleatum, Porphyromonas, Streptococcus that are in everybody's mouth. But if you see those in people's colon or in the stool sample, it's generally a sign of disease, whether that's some Crohn's disease or colon cancer. And so a big question that we don't know, and I'm not going to answer today, is how these oral bugs that everybody has get into the colon of people to then help drive disease. But again, we think that the role of the microbiome is somehow into driving inflammation to increase cell proliferation combined with the earlier mutation to drive tumorigenesis. And so a former student of mine, Joe Zacular, who many moons ago came and taught a workshop with me here, moved to Michigan, used a mouse model of tumorigenesis, where we treat mice with azoxymethane, which induces a mutation, and then multiple rounds of giving them sodium sulfate in their drinking water, which induces inflammation. And so with this model, mice will develop tumors, say 20 tumors or so, colon, over the course of like 2 months. Okay, so it's very fast. It's a model. It's fairly artificial. But what Joe was able to show was that over the course of this model, the community changes. And if he takes those mice running through the model and treats them with different combinations of antibiotics, he can change the number of tumors that the mice would get. He also did other work that's reported in mSphere from a number of years ago to see that different types of bacteria are showing up in these communities, and that if he takes a community associated with large numbers of tumors, puts that into a germ-free mouse, those germ-free mice will get more tumors than if the germ-free mice got stool from conventional mice. And so we're fairly convinced that there's something in the microbial community that's helping to drive tumorigenesis. And so we're thinking about this again from a community perspective and thinking about, can we use this information that our knowledge of the microbiome is at least associated with changes in tumor progression to think about making a diagnostic. And so again, working with Mac Ruffin, who's now at Hershey, and others at the University of Michigan, we got access to the EDRN, Early Detection Research Network, Great Lakes, New England 007 study. So this is a clinical cohort where they recruited close to 500 individuals with varying tumors. So about 170 had no lesions in their colon, 318 had a lesion, about 200 of those had an adenoma, and 120 had carcinoma. And we knew their colonoscopy result. So from this, we also knew whether they had signs of blood in their stool. And then from this, we've taken these stool samples and we've done a bunch of multi-omics approaches on these. 16S, which I'm largely going to talk about today, we've done shotgun metagenomics, shotgun virome data, and we've also done some targeted metabolomics looking at things like short-chain fatty acids. And the general question is, can we differentiate between people with and without lesions? So if I have a stool sample, can I predict whether or not that person has a lesion? And so again, what I'm going to focus on with you today is talking about our 16S work, and maybe next time I'll have more information to share with you on some of these other things with virome and metabolomics. And so one of the things that microbiome researchers love to do is make an ordination, to make clouds of communities. Where on this plot, the red dots are lesions and the blue are people with normal colons. And we look at their community structure, and the distance between these points tells you how similar or dissimilar two communities or two people are from each other. And when we look at this, there's no obvious community-wide difference between people with and without a lesion. So there's no just blazing signal to say that there's a colon cancer type microbiome. And so this is a thought that then maybe there are sub-communities or groups of bacteria that would allow us to perhaps differentiate between people with normal colons and those with lesions. If we look at something like diversity, like kind of our backup trick, looking at alpha diversity, we see people with normal lesions have, it appears to be slightly higher diversity than people with a lesion, but it's not statistically significant. If we look at the specificity and sensitivity, it's not predictive whether or not high or low diversity is predictive of whether or not someone has a lesion. So this isn't super helpful. And if we do the trick where we look at all the possible taxa in the gut microbiota and then say, are there any bacteria that are positively or negatively associated with colon cancer, we find that when we do this, we find two bacterial populations, one related to Porphyromonas, one related to Peptostreptococcus, that appear to be higher, significantly higher in people with lesions than those without lesions. So the question is, that's cool. Can we perhaps use these as predictors of whether or not someone has a lesion in their colon? And so over on the right here, we have another one of these curves drawing the specificity with the sensitivity. So there's a trade-off. You can detect everything, just really detect things with sensitivity, and then whether or not you're overcalling things, which is like the specificity. And so if we have this, we have a long 45 degrees, that's basically what you might get by flipping a coin. And so, yeah, we do a little bit better if we knew whether or not people had Porphyromonas or Peptostreptococcus. But this isn't like a great signal for telling us whether or not somebody has a lesion. And so what this tells us is that, sure, these two populations are probably important in differentiating between people with and without lesions. But it doesn't tell us the whole story, because not everybody with cancer has Peptostreptococcus or Porphyromonas. But there's a lot of people with lesions that don't have these organisms. And of course, then there are some people that have these organisms in lower abundances. And again, I see people do this type of analysis using tools like LEFSA or Metastats or t-tests or whatever your favorite statistical test is. But it's really looking at the community, like population by population. And again, we're looking at a community, right? So we want to be able to look across many populations to see if there's mixtures of populations that are indicative of disease. And so when we do this type of thing, we're really going back to kind of the Koch's postulates, right? What is the bug causing disease, rather than what are the groups of organisms causing disease? And so this gives rise to a set of techniques that we've really become fond of, as has like everybody, you may not know this, but machine learning tools. And so I think of machine learning and a tool that we use a lot called random forest as a spam filter. And my students outed me for being dated in my examples of spam. So I think the modern thing that people worry about are phishing attacks, right? And so perhaps you have your own built-in filter of things you look for to know whether or not someone's phishing, right? So if someone asks you to like email them your like password, right? Or if it's from an email address that's like psu.edu, so if it's pus.edu, right? You that doesn't look good, right? That's from a phishing attack or something. The example I think of in terms of spam is, an email coming from a Nigerian prince that wants to give you $1 million, right? There's all sorts of signals in there that scream to me like, this is not real. I mentioned I have a farm. I've been getting these emails from someone in Saudi Arabia that wants to buy me land and help me raise 1,000 sheep, right? Like, this person knows a lot about me and what it would take for me to like quit my day job, right? So but if like comes and says $1 million, like maybe that's from NIH, and I want to know that. So $1 million on its own isn't useful. Nigeria, maybe like the big outbreak of colon cancer cases. I want to know about that. But when you like start combining these features together, that sends off all sorts of signals that this is spam. And thankfully, if you use like Gmail or your university email accounts, they've got these built-in spam filters, and so we don't see a lot of this stuff. And so that's what we're trying to build with the microbiomes. Can we build a cancer filter? So that if I can look at someone's microbiome and then say, hey, you've got a lot of parvomonas, do you also have fusobacterium? If you have a lot of fusobacterium, then we're going to say you have a lesion. If you don't, then we'll say maybe you're healthy, right? And so we can build these trees that are a lot like, I don't know, back when I was a kid in biology class and you had to figure out, is this a maple tree or an oak tree, right? There's all the things you look for about the leaves or the bark or the height or the canopy size. And we're kind of doing the same thing with the microbiome, where we're trying to classify a community as being from someone with a lesion or having a normal colon. And so what we find is that, again, if we think about classification of people with normal colons versus those with lesions, that if we build these classifications that take into account the whole community, instead of getting a line like we saw for Shannon, or those Porphyromonas or Peptostreptococcus that are kind of hugging this 45-degree line, we do a better job of classifying individuals as having normal colons or colon cancer. And so the metric we like to use is AUC, which is the area under this curve. So if AUC is 0.5, it's basically random. If it's 1, then we probably think we've done something wrong because nothing is that perfect. And so 0.7 is pretty good, but we'd like for it to be better. And so the take home from this, again, is that instead of just using one organism to classify people, if we then take all of the bacterial populations in the community, can we then improve our ability to classify people as having healthy colons or lesions in there? And so what are the bugs that are driving this? Well, it's a lot of stuff. Right? So I think the 20 or so, maybe 18 populations of bacteria, we call them OTUs, that show up in our model. And red are people that have lesions, the blue are people that have normal colons. And you kind of see things all over the place. And so it's really hard to see anything that really pops out. Perhaps this Porphyromonas sticks out to you. And I think what happened is that Porphyromonas behaves a lot like the Peptostreptococcus. The other thing that jumps out at me is that with one exception, this Bacteroides, there isn't an OTU or population that is found in everybody. So everybody is very different. And so there isn't something that really jumps out as being a bug or microbe that is driving disease. And so one of the things that I've been playing with is like, how can we visualize this better? And so hopefully this doesn't induce seizures. But this is the blue line. Iteration of the line represents a different individual that had a normal colon, and the relative abundance is across these 20 populations. And so what you can hopefully see is that there's many ways to be normal, to have a normal colon. And similarly, if we look at people with lesions, there are many ways to have a lesion. And so the beauty of these random forest and machine learning techniques is that it kind of sifts through all this complexity to give a classification of being normal or having a lesion. And again, this gets us back to thinking about these polymicrobial traits, that there are many taxa, or many genes, if you will, and there are many abundances, or alleles, if you will. And we can, by mixing and matching those, biology sifts out, and then you either have a normal colon or a carcinoma. And so, can we use this information to think about a diagnostic and to think about risk? Max strikes again. This is using FIT, which is the fecal immunochemical test. And so the normal over here, so typically people don't have blood in their stool. And if you have a lesion, you're more likely to have blood in your stool. And so in the United States, at least, the threshold for suggesting someone get a colonoscopy is it's about 100 nanograms per mil. And so these people up here would be encouraged to get or I guess these people over here would be encouraged to get a colonoscopy. And so you can see that the test is fairly specific, but kind of low in sensitivity, especially for a lot of early stage tumors. And so the AUC here is 0.75, which is better than what we saw for the microbiome alone. And so a former student in my lab, Neil Baxter, said, well, can we take the microbiome results, FIT results, blend them together and get a better model? And so, okay, so this is the microbiome, and then if he adds them together, we see in this orange-yellow line that we see an improved AUC because we're taking kind of the information along with the microbiome information to get a better sensitivity. And if we then add metadata, so say like if we know the person's age, their BMI, their sex, we can perhaps do a little bit better as well. And so by adding all this extra information, context, clinical information, we can improve the ability to make a diagnosis. The next thing that Neil did was to say, okay, this is what these curves look like for sensitivity and specificity. He wanted to focus on this yellow line. And so if you picked a threshold of, I think it was 80% specificity, what is the sensitivity? 90% specificity, what's the sensitivity? And he looked at people that had lesions classified different ways. The white bars or gray bars are FIT alone, and the black is using FIT plus the microbiome. And so what you'll hopefully see is that for these earlier stage tumors of adenomas, we did a significantly better job of detecting adenomas than FIT alone. So again, this gives us hope that we could use the microbiome to do a better job of non-invasively diagnosing somebody as having an adenoma. And what we find is that if you've got a carcinoma, that FIT actually does a really good job of detecting carcinomas. And so adding microbiome information doesn't improve the detection. So the microbiome doesn't do everything. Right? We've got something else to do that. And also like the number of individuals that we have with these higher progressed tumors is relatively low compared to what we have for adenomas. And so there might be cases where FIT fails that we might be able to supplement with more microbiome. One of the things that another postdoc in my lab, Mark Z, did was to take data from Neil's work, and about 60 or so of the subjects that had carcinomas, we had a follow-up sample on after the person had been treated. And so what we have in this graph here on the left is the probability, according to the model, of whether or not the person had a carcinoma. And then after it was resected and they were treated, that we basically saw a significant reduction in the probability that the person had a lesion according to our model. So our model is telling us that these people are becoming more normal after treatment, which is cool. One of the things that sticks out to us is that there's a few people that their probability went up after treatment. And because of limitations of IRB protocols and subjects research, and these are actually humans and not mice, is that we don't know what happened. Right? And so we wonder, we hypothesize that perhaps these people had recurrent disease. And so we don't know whether or not they did, but it would be great to be able to think about these studies more long-term in terms of following up and seeing, can we perhaps predict who's going to recur based on their microbiome or after treatment. So context matters. We've shown that, I think, that thinking in terms of the context of the entire community of bacteria matters. It's not just one organism that we think is causing disease, but we also know that things like sex and age and other comorbidities matter, certainly for detecting a lesion, but we think also for helping to drive the disease. We can also imagine that things like host gene expression, the person's diet, and other factors might also be important in making a diagnosis or in driving the disease. And we kind of hope, in a way, that by looking at this from a diagnostic perspective, that will also give us leads in thinking about things perhaps more from a mechanistic approach, right? That if we understood, okay, these oral organisms keep popping up, why is that, right? What is it about the oral community that gives rise? Is there a link between periodontitis and colon cancer, Crohn's disease, where we also see a lot of these organisms popping up? And so I'm going to shift a little bit because at this point with the colon cancer stuff, we don't have a lot of kind of more reductionist mechanistic type of details, and tell you about a story that we've been working on in our lab in collaboration with a bunch of other labs at the University of Michigan, regards to Clostridium difficile infection. So a colleague of mine, Vince Young, and I have a systems biology grant to study C. difficile infections, and have a hospital right down the hall from us that unfortunately has C. diff cases like every other hospital. And so we've been able to get any sample that is submitted to the microbiology core for C. difficile testing. We get those and we bring them into our freezers, and doing everything from community analysis, which is what I'm interested in, to Evan Snitkin and another faculty member is sequencing genomes of all these C. difficile. And doing just a really big multi-omics study of our hospital, but then also a mouse model of C. difficile infection that we have. So again, answer many of these similar types of questions. And it doesn't appear to be there's one bug causing disease, it's a community of organisms. And so if we're looking across a community, can we think about mechanisms across a community? And so if you're not familiar with C. difficile, consider yourself lucky. That about 500,000 people each year are diagnosed with a C. difficile infection, CDI. And about 20% of those individuals, once they're treated for C. diff, will have recurrent infection. So they kind of create this spiral where they're getting treated, and the treatment, antibiotics, makes you more susceptible to disease. So it's kind of smart. So for people over the age of 65, 1 in 11 die of an infection. And people on antibiotics are more likely to get a C. diff infection. And if you work in a healthcare setting, it's likely that you're asymptomatically colonized or at a higher risk of becoming colonized. And so this is a problem for people that are frequently interacting with healthcare. And because there is this link to antibiotics, it makes sense to us that the microbiome is involved, right? So you perturb the gut microbiota, C. diff can come. And I like this also because it gets me to think ecologically. And that if you think about, if you had a forest fire, or if, it gives an opportunity for all sorts of other organisms to colonize that perhaps aren't indigenous to that environment. Or if people thought it was a brilliant idea to bring gypsy moths into the United States, which is like an invasive species, right? And so we've got C. diff, which in many ways becomes an invasive species because we've changed its niche space that it can now exploit. And so these are hypotheses that we've had rattling around as microbial ecologists and trying to think about C. difficile, thinking about the niche space of C. difficile and questions of whether it's a generalist or specialist, and whether or not that's helping its pathogens. And so again, the general model that we have is that people have a normal gut microbiota, they're treated with antibiotics, they're losing a fraction of this community, they're then exposed to C. difficile. Those spores then germinate, and then people get infected. This produces a toxin, they get sick, they get treated with antibiotics. Some people will recover, others will have recurrent disease. If you get on this kind of recurrence loop, the most effective treatment is a fecal transplant, where it kind of doesn't seem to matter where they get the feces from. But if you get a fecal transplant, usually supplied to the patient by like a colonoscopy or an enema, it's like 95% effectiveness. And so it's one of those things that I remember talking to gastroenterologists like 15 years ago, and they would be laughed at conferences for suggesting. And now it's like, Vince Young, who I collaborate with a lot, has gotten comments back from study sections saying, hasn't fecal transplants cured C. diff? Why are we studying this? And so, but again, like, if we think there are diseases transmitted by the community, like say colon cancer or other things, then we want to be careful about who we're giving fecal transplants to and how we're getting. And I liken fecal transplants to kind of like the early days of blood transfusions, right? Like, I can save your life, but shoot, we give you hepatitis, right? And so I'll save your life by giving you this fecal transplant, and you won't get C. diff, but we don't really know the long-term ramifications. There might not be any, but we want to be cautious, and we perhaps want to have a little bit more of a precision medicine approach rather than giving somebody a fecal sample that we don't really understand. And so, yeah, we could give everyone a fecal transplant, say it's over, but there are risks there, and we want to understand even, like, why does this fecal transplant work? We don't really understand. And so, as I said, we've had this big cohort of hospitalized patients. Alex Schubert, a former student of mine who now works at the FDA, actually regulating fecal transplants, had a cohort of about 340 subjects. About half of them were outpatient individuals that had normal stools. 90 were inpatient and had diarrhea, and their samples were submitted for C. diff testing. And about 90 were inpatient in our hospital with diarrhea and had C. difficile. I don't know why I have that result. And so we've, again, done a lot of multi-omics analysis against sequencing, patient medical record screening. We've done culturing and genome sequencing, and all sorts of stuff. And one of the questions was, can we differentiate between people with and without CCL? And now there's a great test that the hospitals use to detect whether or not someone has C. difficile. But the idea of looking at the community was, again, to say, what is it about the communities that are different between people with and without C. difficile? And so this is, again, work from Alex Schubert that was published 5 years ago or so, 4 years ago. And so she was able to take those 340 subjects and to bin them into one of 12 different types of communities. And those community typings were done based on the types of bacteria, based on these dozen or so different types of bacteria in their gut, to see how they arrayed based on whether or not they were a non-diarrheal control, a diarrheal subject, and I think this is colonized with C. difficile. And so we found that people that were high, say in Enterobacteriaceae and Enterococcus, tended to have more likely to have a C. difficile infection. And this type of work is hard because just because two people might have a similar community, one colonized, one not colonized. The person that's not colonized might just not have seen C. diff in the hospital. Right? So for whatever epidemiological reasons, if I'm treated with antibiotics and I never see C. diff, I'm not going to get colonized. And so it makes it really difficult to think about what communities are allowing for colonization because they never get exposed, then we don't know. And we can't do that exposure. Right? Like, we can't give you all antibiotics and give you C. diff. Or not, or just give you C. diff without antibiotics. We wouldn't want to do that. But, skip over this for now. But we can do this with a mouse model of C. diff that we have. And so Alex and later Matt Jenner, who was another student in my lab, worked on a mouse model that Vince's lab has really done a lot of work with, along with others in the field, where we take mice and we treat them with various different antibiotics. We give them a dose of antibiotics, and then we gavage them with C. difficile, say on day 0 here, and then we start watching for colonization. And in this study from Alex's thesis, what we wanted to do is to use these different types of antibiotics and different doses and recovery periods to see if we could generate different starting communities. Right? And so we could generate basically a bunch of different patients in the hospital, but using mice. And then if we gavage those with C. diff, can we predict the number of C. diff that will be in their feces on day 1? So instead of figuring out who's going to get it or not get it, can we predict how many they're going to get? And so we see a lot of different types of bacteria. We see, ampicillin versus metronidazole. We see very different types of community structures. And then we also see, in some cases, with 1 day of recovery from the antibiotic, say 10 to the 7 versus 10 to the 8 CFU of bacteria. And if we let them recover longer, say for 6 days, we see more resistance. And so the goal isn't to make a resistant mouse. We've got that from an untreated mouse. But by using these different antibiotics and doses and periods of recovery, we can generate different levels of colonization. And so what this shows is along the y-axis is the observed colonization for the different antibiotics we gave them. And the x is, again, using random forests to predict colonization. And we basically see that this cloud falls on the 45-degree angle, that we do a pretty good job of predicting colonization based on the key structure at the time of C. diff challenge. And so that was pretty exciting because, again, we could look at the microbial community and predict whether or not they'd get colonized and to what level. Then when we start pulling this apart and looking at the actual bacteria that are there, we see a mess, right? We see a lot of context dependency. And that this, like I said, we're struggling to figure out how to depict this variation and kind of sub-network or sub-communities that are driving disease. But we see cases where, again, if we have high levels of Escherichia, we'll have high levels of C. diff. But if Escherichia is not there, we still can see, in some cases, higher levels of C. diff. Right? And so we see some context dependency where if a bug is present, it might be causing disease, but also if it's absent, we might still see signs of disease. And so depending on what organisms are present in the community, we can get mixtures that then drive the colonization levels of C. difficile. So Matt Jenier took this on and said, well, how can we explain some of this context dependency and variation in the community? And so Matt picked 3 of the antibiotics, streptomycin, cefoperazone, and clindamycin, and sampled the ceca of these mice after 18 hours and wanted to look at gene expression in the community and the gene expression of C. difficile. And he wanted to know what's C. difficile doing in these 3 different conditions, as well as in germ-free mice challenged with C. difficile, and then also what is the community doing under these conditions. And the hypothesis was that C. diff can adapt its physiology, its gene expression, to deal with the communities that result from these different antibiotic treatments. And so, sure enough, with different antibiotic treatments, we see different levels of colonization, varying levels of toxin production. Mice that were free, that were colonized with C. diff, get a lot of toxin, a lot of colonization, and we see kind of mixed results from those other antibiotics. We use a kind of wimpy strain of C. diff, strain 630. We don't want to use a really aggressive strain because then the mice stop defecating and can't do our work. But kind of looking at the intermediate pathogenicity phenotype, we can look at kind of the progression of the disease. And then this is, people that do soil science are perhaps familiar with this type of plot, a ternary diagram that allows us to plot gene expression for C. difficile on cefoperazone, streptomycin, and clindamycin. The size of the circle indicates the number of transcripts that Matt saw from C. diff, and the color, or the placement, indicates how it was enriched in each of these 2 different conditions. So these green ABC sugar transporters were more enriched in cefoperazone-treated mice relative to the others. And then there's, of course, a bunch of stuff in the middle and stuff that's more pronounced in cefoperazone and streptomycin-treated mice that are absent in clinic. Right? So again, we've got this very multifactorial data that's challenged to represent. We're always kind of struggling with how to do it best. And so what Matt found, again, with ABC sugar transporters, when C. diff goes into a mouse treated with cefoperazone, it produces a lot of gene transcripts for ABC transporters. On clindamycin, a lot of disaccharide metabolism genes, and then sugar alcohol metabolism seems to be more pronounced in streptomycin and cefoperazone-treated mice. So supporting our hypothesis that C. diff is able to change its gene expression to deal with the community that's going into, which leads us to believe and assert that it's a generalist that can really do whatever it needs to do to exploit open niches because of these antibiotic perturbations. And so with that, I'll stop. There's more I could tell you, but hopefully giving you a taste of how we think about the role of the microbiome in health and disease. Whenever I give these talks, I really want to give a big shout out to the patients that have participated in these studies. we lose track oftentimes when we look at those ordinations of those red points, that is a person who's been diagnosed with colon cancer or adenoma, and that they're brave enough to agree to enroll in one of these studies is a huge thing. I don't know that I would be willing to do that. And so really appreciate their contribution. And then all the great people and collaborators that we've had the opportunity to work with. As you think about the center that you have here, and talking with Carol Lee and others, that there is a lot of collaboration that, sure, we've got people from microbiology and immunology in my department, in my lab, but we also have people like Matt Ruffin, who's a great clinician, and Elena Stoffel, who's a clinician, Kim Turgeon, Grace Chen, who are all great oncologists who study this. This is what they do. And then I come along, and I'm the microbiologist, and they've got to teach me, and I've got to teach them. And then I'm working with epidemiologists and statisticians like Ray Rogers and Jenna Wines, who do a great job of helping us to kind of think about the multidimensionality of our data, trying to get us to a result of whether or not a person has a lesion or not. So with that, I'd be happy to take any questions people might have.
So I think that's the composition of the community is more important than the experience.
Yeah, so in our analyses, when we've done like qPCR to try to quantify the number of bacteria, even like antibiotic treated mice, that by the time we start looking at them, we don't see significant differences in the density of the communities. But I think there's certainly diseases where that might be a factor. So work with a colleague, John LaPuma, who studies cystic fibrosis. And there we wonder a lot, is CF being driven by density or specific organisms? Or it's probably both. Never so simple as one or the other. But at least in our context, we don't see big differences in density.
Great talk. I'm not familiar with machine learning, but it seems like it's something that is very useful for looking at fluctuating data. How exactly is the machine learning to distinguish normal from abnormal? Like, is it being told different factors to look at, or is it just noticing this is distinctly different?
Yeah, so I don't want to diminish what machine learning is, but if you've ever done a linear regression, that's a form of machine learning. If you've done a logistic regression, that's machine learning. If you've gone into Amazon and wondered, like, wow, how do they know that I want to read that book or want to buy this coffee maker? That's machine learning. And so what you do is there's a training phase where you say, okay, all these people have colon cancer, these people have normal lesions, or normal colons, right? And this is the information that we want you to use to differentiate these pools. And so you train it by saying initially, like, this is a control and this is a case, and then through its magic, and there's many algorithms to do the machine learning, it can sort it out.
So then does it move on to pick out other things that you don't tell it?
Yeah, so because I could approach this and say, so I knew like it was probably important to differentiate, but when it came to like the microbiome, I might not know, or there might be populations I think are important, and then it can go in and say, yeah, those might be important. These other things are also important, and then again, it thinks about it in a compositional nature. So it's not just thinking about one thing, but kind of combinations of things.
I'll just add, we have some people here in the Microbiome Center and state that are experts in that area, if you're interested.
The question, when we have sequencing, sort of everything is relative to the total amount of DNA that we extracted. What if, so is there any absolute value? Do you think that matters a lot?
Yeah, I mean, that kind of comes to this of like, how important is density? So I think it probably does. I think it does matter. For things like the colon, where it's so dense with bacteria anyway, I don't know how much it matters. And I think for thinking about mechanisms of disease, that's probably a good question. For something like diagnostics, I don't really care. If I can detect a signal, that's great. I was at a meeting with NIST, the National Institute of Standards and Technologies, and so they want to know, like, what does the microbiome really look like? So whenever you do this, you do a PCR step. PCR skews everything. You don't understand how. And so the people at NIST are all kind of bent out of shape, like, we got to standardize how we do this. And I said, I don't really care about the truth. And they all laughed like you all did, and they're like, get this guy out of here, stat. And I was like, because of my assay, if my flawed bias assay gives me a good classification, I'm good to go. But perhaps adding something like bacterial density would be important as another feature to think about classification. At least from what I've seen with stool, we don't see a lot of variation. Could be something to think about. But for like a mechanism.
Question. For example, you have a bacteria becomes very, very abundant. This patient may have more Clostridium difficile than the previous patient. It's just they have another unrelated bacteria that makes it look that might much less
Sure.
than what I have.
Yeah, so are you saying like, so there's certainly, are you thinking about like density in terms of severity of disease in that case? Or?
You know, when you sequence the sample from what DNA you have in there, and you can only say when you do percentages, right? I'm just thinking about, so I always have this
Sure.
question when we observe something, whether it's reality or it's just this filter of what we were able to extract and see.
Yeah, so it's definitely through the filter. And so what we're saying is that the filter is okay. We can live with that, but there's probably more to be gained by trying to strip away some of that filter, right? So moving to something like qPCR, or if there's other people played around with kind of spiking in a control where we know the abundance and we can kind of work out those types of things. That's probably something on the way, but we're kind of all into this mode of extracted, amplifying sequence.
So I'm going to end us there. I just want to say Pat did an excellent job in advertising for next week's session. If you haven't been reading the Microbiome weekly newsletter, Matt Ruffin and many of the colleagues from Hershey are going to be up here next week, and we're really trying to put together some interesting cross-disciplinary collaborations. You want to talk to them, figure out what they're doing, how you can work with them, what ideas they have that might relate to work you're doing and trigger new observations. So, excellent opportunity. Please sign up today because we're trying to figure out lunch.
And I would underscore, like, so 9 years ago, before I moved to Michigan, I never worked with a clinician, and it's totally changed how we think about our science. That I can design all sorts of mouse experiments, and I'll go pitch them to Vince Young, whose office is right next to me, and we would exchange crazy ideas. He's like, "You'll kill people," right? Or like that. We would never do that, or we'd never think about that. Or working with Mac, you know, I say things like, "Can we replace FIT?" Or maybe tell who doesn't need a colonoscopy if they have a positive FIT. And it's like, no, because FIT is just part of what we do. That you'll never get away from FIT. So that's got to be part of the algorithm. And so you really get a context of working with these people. And my trainees have all had the opportunity of now working with clinicians and seeing a colonoscopy get done, or working, got a cancer genetics group, and seeing them meet with families and talking about diagnoses, walking the halls with Vince diagnosing C. diff. And it's, you might think it's like not that big of a deal, I'm just a bench scientist, but it really gives, you know, added texture to your treatment. But definitely suggest you do that.
Let's thank Pat. If you haven't signed in, please do.
Have you seen